Alright everybody, and welcome back again. This is lecture 3 still. This is part 2. I apologize for the delay. We were having some technical issues earlier in the week, about midweek when I wanted to post this, but here it is. Again, this information is still in your textbook. Most of this slide, compared to the last part 1, is actually taken from your book as well. So we're still covering cell membranes, enzymes, energy, and our focus on this part 2 is the actual organelles themselves. So I started introducing you to the organelles. I started telling you about some of the key features of things like the nucleus, the endoplasmic reticulum, ribosomes, things like that. So that is what I'm going to go into detail about here with part 2. So again, eukaryotic cells are partitions. Remember, the cells have multiple copies of, well, all cells have multiple copies of various organelle structures. Depending on what the cell's specific function is, it could have hundreds of mitochondria and millions of ribosomes, for example. Each of these organelles is bounded by a membrane with a lipid and protein composition that suits its function. So, the cell itself contains and is made up of a plasma membrane, which remember is composed of phospholipids mixed in with proteins, our fluid mosaic model, if you will. And then when you go inside the cell, within the cell itself, each of these organelles has its own membrane bound system as well, also composed of phospholipids, proteins, etc. So that's what this is targeting and telling you about. So the four basic functional groups within these partition cells or within the organelle system is the nucleus and the ribosomes carry out the genetic control of the cell. So remember, the nucleus is where our DNA, our instructions on all the proteins that we make in our lifetime is basically held. And it's the ribosomes, which is composed of, remember, ribosomal RNA and is also made up of some protein, and they are made up of two different parts, and we'll see the ribosomes in detail in a couple of slides coming up. But together, they work together, right? So the nucleus carries the DNA gets transcribed into messenger RNA, that messenger RNA hooks up with the ribosomes, could be out in the cytoplasm, for example, and the ribosomes translate the DNA into protein. Organelles themselves are involved with the manufacturing, the distribution, and breakdown of molecules. Specifically, what we're looking at is the ER, or the endoplasmic reticulum, the Golgi apparatus, followed by the lysosomes, vacuoles, and peroxisomes. Now remember, the ER and the Golgi, this is where once proteins are starting to be, say, put together out in the cytoplasm by ribosomes, a lot of these proteins will get transported to the ER, for example. So when they are taken to the ER, they are going to be further processed. Okay? They're going to be, well, just like an emergency room. Perfect example. That just came to me. So when you go to the ER, they assess you, and they're either going to repair you or they're going to send you on your way. The endoplasmic reticulum is basically the first stop for a lot of proteins. So once they are made either out in the cytoplasm or as they are being translated, they will sometimes be taken to the ER. The translation process will finish and continue, and that protein is directed into the ER already, the endoplasmic reticulum. And within the endoplasmic reticulum, what we'll see a lot of times is that proteins are dressed up. You know, they might have other proteins or other features added to them. They might undergo different types of folding processes might occur within the ER. And then, of course, a lot of times what we'll see is proteins then get shifted and taken to the Golgi apparatus. And it's in the Golgi apparatus where proteins, for the most part, I would say, majority of the time, are then enhanced in the Golgi. So a lot of times they'll get sugar molecules added to their protein side chains, for example, and they become known as glycoproteins. And then ultimately, sometimes towards the end of a protein's life cycle, if you will, they end up going to lysosomes or the vacuoles or peroxisomes, which are also membrane-bound structures in the cell, but their environment inside of them, so for example, the lysosome, is going to have a much higher pH or a much lower, pH level than, say, the cytoplasm. And this is because in the lysosome, we want to break down the proteins back into their primary elemental stages of amino acids again. Okay, so keep these key things in mind here. The mitochondria is in all cells. Chloroplast is the plant version of the mitochondria, and they function in energy processing. Again, next week we are really going to hit it hard with cellular respiration and discussing what this cellular respiration process is and how is it that we are able to generate all the energy within our cells, within our bodies, to do what we do on a regular basis and beyond. And then, of course, ultimately what we're also going to touch on, at least here in this lecture, is structural support. So the movement and communication that happens between and among cells happens via the cytoskeleton, the plasma membrane, and with plants, the cell wall. So the nucleus, again, contains all the cell's genetic instructions encoded in DNA. Understand basic features of DNA, please. What is DNA? Deoxyribonucleic acid is what it stands for. Understand that it is made up of adenines, thymines, cytosines, and guanines. A, T, C, and G. A always pairs with T, and C's always pair with G. So basic things like that. It's a double helix. DNA is associated with many proteins and organized into structures called chromosomes. So remember, when the cell is not dividing and the cell is intact, it's at rest, whatever the case may be, the DNA is housed in the nucleus and it's wrapped around DNA. DNA, anytime it's associated with proteins, we can call that structure a chromosome. But specifically when the cell is not dividing, and again is at rest now, the combination of the proteins that coil up the DNA and keeping it together, this feature, this structure is called chromatin. Okay? So, and then surrounding the nucleus, its membrane is a specific name called the nuclear envelope. So the nuclear envelope is the double membrane system that encloses the nucleus. Okay? And each of the two membranes is a separate phospholipid bilayer, also with associated proteins. And many of these proteins within this nuclear envelope do various things. They support the structure of the membrane and maintain the integrity of the nuclear envelope. They also help with shuttling DNA and proteins in and out of the nucleus during the transcription and translation process as well. The nucleolus is a site within the nucleus where the ribosomal RNA is synthesized based off of instructions in the DNA. These proteins are brought in from the cytoplasm, are assembled with the ribosomal RNA, and they form the subunits of the ribosomes. Okay? So again, the nucleolus is the site where ribosomal RNA, which ribosomal RNA is synthesized based off of instructions in DNA. Then you have proteins out in the cytoplasm, so outside of the nucleus, that are assembled and come in and will hook up with the ribosomal RNA So proteins already made plus the combination of ribosomal RNA is what makes a ribosome. And then ultimately, remember, messenger RNA is basically a set of DNA instructions for a protein that have been transcribed into a different language, messenger RNA, and this messenger RNA is what the ribosomes read and then translate into protein. Again, please review some of these key YouTube videos that I am posting specifically about protein synthesis. It's extremely important and it's very crucial. Okay, ribosomes themselves, again, machines that carry out the commands of the nucleus. They are cellular components that use the messenger RNA to build the protein. So in this image here, for example, your ribosomes are made up of two subunits, a large subunit and a small subunit. And when they come together, they're a fully functioning ribosome. And what's happening here is the messenger RNA is being passed through the ribosome. And as the ribosome reads the messenger RNA code, it will then attract and bring the amino acids into play here. And remember, the amino acids are brought into the ribosome through a structure known as tRNA. Okay? So tRNA basically carries the amino acid and brings it into the ribosome, and it's within the ribosome that the amino acids hook up together and they start generating a polypeptide chain, and a polypeptide chain of amino acids ultimately will become a fully functioning protein. So again, understand that process, rewind these last 30-45 seconds and replay it as often as you can. This is clearly a very important feature, people. Other thing too, right? You've got free ribosomes versus bound ribosomes. So free ribosomes are ribosomes floating in and around the cytoplasm as messenger RNA comes pouring out of the nucleus. The ribosomes capture these messenger RNAs and the process of translation occurs. Then there are times where ribosomes are already attached and bound to the endoplasmic reticulum. And that's where we get the term rough ER, which is coming up in a little bit, because it looks rough in a microscope, the ER, but it's simply the portion of the endoplasmic reticulum where ribosomes are already attached to. Okay. The endomembrane system. So internal membranes, again, a distinguishing feature of eukaryotic cells that are involved in most of the cell's function. Some of the membranes are physically connected, and others are linked when tiny vesicles transfer membrane segments between them. So what's that mean? Okay, so in essence what we're saying is, again, just that some of the membranes, so for example, the endoplasmic reticulum and the Golgi, those two systems, organelles, each have their own respective membranes, but they are not connected to each other. But proteins are transported from the ER to the Golgi via transfer vesicles or vesicles. So vesicles simply are just that. They're these little structures that form where a protein gets packaged, and part of the endoplasmic reticulum membrane actually pinches off and forms its own little bubble, if you will. And within this bubble contains some of the endoplasmic reticulum membrane, as well as the protein it's transporting. And this bubble moves onto the Golgi apparatus, where the protein is deposited into the Golgi for further processing. Okay. And again, this endomembrane system where you have some organelles with membranes attached or not attached. It can be the nuclear envelope, the ER, the Golgi, lysosomes, and the ultimate plasma membrane. Remember, the plasma membrane is the lipid bilayer membrane protein system of the entire cell. Okay, again, just reiterating, the endomembrane system is the largest component of the system in the ER. The endoplasmic reticulum, the ER, is an extensive network of flattened sacs and tubules, and these tubules and sacs of the endoplasmic reticulum enclose a space that is separate from the cytosol. So the cytosol is basically everything else that's not an organelle. It's the environment within the cell that is not part of or within an organelle. And then again, within the ER, you have two types. You have smooth endoplasmic reticulum and you have rough endoplasmic reticulum. Flipping back to smooth. Enzymes of the smooth endoplasmic reticulum are important in the synthesis of lipids, fats, oils, phospholipids, and steroids. Liver cells, for example, have large amounts of smooth ER that help process the drugs and alcohol and other potential harmful substances. And the storage of calcium ions, for example, in muscle cells. So in muscle cells, there's a specialized smooth endoplasmic reticulum membrane pump that pumps calcium into the interior or into the empty space of the endoplasmic reticulum. And this is something that, again, we'll see a little bit about later on. But basically during, for example, muscle contraction, when that is stimulated to occur in the body, we require calcium ions to be present in order to activate proteins in our muscle cells to actually do the contraction. And again, the smooth endoplasmic reticulum is needed, or is the type of endomembrane system that we want in place for things like this to occur. Then you have the rough endoplasmic reticulum. Again, this is part of the ER that if you look under a microscope and you're looking at an ER or an endoplasmic reticulum structure and it looks rough, or it looks like there's little things studded around it, that's the ribosomes, right, attached to them. So basically many types of cells secrete proteins produced by ribosomes attached to the rough ER. And insulin is a perfect example. So we're going to see this mechanism in just a minute, but basically what happens is, let's talk about insulin. So insulin, remember, is a protein that's needed to help take glucose out of our blood and bring it into our muscle cells for storage purposes, ultimately. But the way insulin gets made is like this. Real brief, real concise explanation here. Insulin goes through the standard translation process. The DNA code for insulin gets transcribed and translated, and as the translation process is occurring for insulin, the ribosome will not make insulin out in the cytoplasm. The ribosome will start translating the insulin protein in the cytoplasm, but maybe about halfway through, it will take the rest of the messenger RNA that is encoding for insulin, and it will take it to the rough ER, and that ribosome will bind to the membrane of the endoplasmic reticulum. Translation will reactivate, and as that insulin protein is made, it is now being fed or threaded through the ER membrane and dumped into the endoplasmic reticulum. Once it's there, short chains of sugar are linked to the polypeptide, making glycoproteins. This does happen, for example, mostly in the Golgi apparatus. and then ultimately when the molecule is ready for export from the ER, it is packaged into transport vesicles. Right? So again, a brief kind of fairly detailed at the same time, but concise explanation of, again, what ribosomal RNA, what rough ER does. Excuse me. Okay. And again, so here is that mechanism. So again, this is the mechanism that, for example, insulin goes through. I could ask, for example, a fantastic test question, and I might say, how does insulin become insulin? How does it go from, being encoded in the DNA and being processed and exported out of the cell? The explanation I just gave a few seconds ago, along with these next 2-3 slides, is going to be crucial. So, again, this image is taken from another textbook that I used to use. It's on the book is one of the supplemental materials that I recommended. But I'm going to point out the key features that I want you guys for this class, for this semester, to understand and to get out of it. So again, not everything on here you need to know in great detail, but I want you to understand what is happening. Okay. So first things first, let me tell you some of these definitions. So SS is the signal sequence, and SRP is the signal recognition particle. Okay, so those are some key abbreviations to understand for this process. And so what is happening here, for example, is we have a ribosome here in the gray, and we have here our string of messenger RNA in this darkened purple maroon-like color. So what's happening is at step 1, the messenger RNA is going through the ribosome, and the ribosome is doing its job. It is starting to translate the protein for us. So it's translating messenger RNA into the polypeptides of amino acids. And that's what's coming out here in blue and pink. Okay? So these blue and pink pearls here, if you will, is the emerging future protein. This is a string of amino acids. What will happen is that this string of amino acids will contain a specific sequence or a combination of amino acids that are emerging. And this significant type of sequence, now again the sequence can vary. It could be, let's say, it could be serine, threonine, and aspartic acid. Okay? And when that sequence, for example, emerges out of the ribosome, that is known as the signal sequence, because what happens then in step 2 is that a protein out in the cytoplasm called the signal recognition particle will recognize that specific sequence. So in our example, serine, threonine, and aspartic acid, which, if you think about it and you remember your one-letter amino acid codes, serine, threonine, and aspartic acid make STD. Okay, so that's going to be our fun little signal sequence there. So what will happen again in step 2, the signal recognition particle will recognize the signal sequence, in our case STD, and it binds. So the signal recognition binds to both the ribosome and to the signal sequence. And the reason that the signal recognition particle is binding to the ribosome is to stop or put translation on pause. And the reason it does this is because in the following steps here, step 3 now, the signal recognition particle is now going to guide the ribosome and the messenger RNA, and it will take it to the endoplasmic reticulum membrane. So it takes it to the ER membrane, and it helps dock or put into place the ribosome actually onto the ER membrane. And the ribosome is going to bind to a specific protein structure that is embedded in the ER membrane. This protein structure is called the translocon. So, the signal recognition particle binds to the ribosome and messenger RNA. It takes it and brings the ribosome to the translocon, which is located on the ER membrane. And when this happens, some GTPase activity occurs. GTP is just another energy currency like ATP. But again, you don't need to worry about that so much. I want you to just understand the process. And so what happens is that once the ribosome docks to the translocon, the SRP, or the signal recognition particle, detaches and leaves, and then this causes the translocon to open up. Translation of the ribosome is reactivated, and now the protein, as it is continued to be translated, gets fed and is deposited into the endoplasmic reticulum. So again, that is the overall process of what I want you to get in this mechanism. This is known as co-translational insertion. So again, step 1, real quick. Ribosome starts to translate the messenger RNA. A specific sequence known as the signal sequence, a string of amino acids, is emerging from the ribosome. That signal sequence is then recognized by the SRP. The SRP binds to the messenger or to the ribosome and to the signal sequence. This prevents and stops further translation temporarily. The SRP brings the ribosome to the ER membrane, and it docks it onto the translocon, a protein tunnel, if you will, embedded in the ER membrane. When the SRP releases from the ribosome, translation is reactivated and the protein is made and fed into the endoplasmic reticulum. Replay the last 2-3 minutes over and over again. Certainly the night before the test would be my recommendation, because I will definitely want to see you guys explain this, and to be able to explain this hopefully as well, if not better. Because again, this is just a very key and crucial feature that happens in the cell. Alright, so that was the ER, that was some of the nucleus, the Golgi apparatus. So again, the Golgi apparatus was discovered in 1898 using a light microscope and a staining technique done by the scientist himself, Camillo Golgi, hence the name, Golgi apparatus. The number of Golgi correlates to how active the cell is in secreting proteins. So again, if there is a specific type of cell, so let's say the pancreas, which secretes insulin, the pancreas cells are going to contain a lot of Golgi, because insulin is a protein that gets made and processed through the ER and ultimately the Golgi, and it's from the Golgi that insulin then gets released out of the entire cell itself. So that would be another example. Pancreas cells would contain lots of Golgi because insulin is secreted regularly and often. It serves as a molecular warehouse and processing station. So that is, one side of the stack receives the transport vesicles from the ER. Vesicles will fuse with the Golgi, adding some of its membrane and contents. The products of the endoplasmic reticulum, or the usually proteins, are modified. And then on the opposite end of the Golgi is a dispatching side, where again vesicles are used to package the protein, the vesicles will bud off of the Golgi and get transported to other sites. Typically, they will get exported, these vesicles, to the plasma membrane to be released from the cell itself. And this is, again, an image showing just that. So, for example, we have proteins being made here on the rough ER. It's rough because it's studded with ribosomes. Proteins are being translated and fed into the ER. And then these proteins get budded off and transported via these little bubbles here, which are the vesicles that I've been talking about. And these transport vesicles will end up going through the Golgi apparatus. And then the protein gets deposited and it undergoes folding in the different Golgi apparati. So the Golgi apparatus is made up of the cis Golgi, the medial Golgi, and the trans Golgi. So this entire thing here is known as the Golgi apparatus. But basically, the protein just goes through different foldings, and again, things like sugars are added at various stages throughout the Golgi. But each time the protein goes through some type of processing and goes into the next part of the Golgi apparatus, it always gets carried and packaged into these vesicles. Okay? And then ultimately, the cells will leave the Golgi apparatus and will ideally, for example, with our insulin example, that vesicle will be taken to the plasma membrane where it fuses, and then in hopes to release then that insulin or that protein out of the cell. Lysosomes, okay, are again a membrane-bound compartment within the cell, and it's known as a digestive compartment because, again, in the lysosome, it's basically where proteins go to die. Once they're done, once they've been used up, or they've been made in excess and we don't need as much of the protein as we thought, those proteins will basically get transported to the lysosome, and within the lysosome there contains digestive enzymes, and usually it's also a lower pH, or it's a more acidic environment. And basically what it's going to do is it's going to basically break down the proteins back down into their basic amino acids. And then those amino acids can be recycled and used to make new or other proteins later on. The mitochondria, again, is the area where cellular respiration occurs. Cellular respiration, remember, in essence, is basically the breakdown of how we break down the food that we eat, and the oxygen and the breaths we take, and how all this combines together to help generate chemical energy within our bodies in the form of ATP. So this is going to be stressed and talked about, and will be our focus on week 4. Okay. So we're not going to go into great depth, but again, I want you to know what the mitochondria does in basic terms right now. Again, it's where we convert chemical energy of foods into chemical energy of the molecules, ATP. It is also enclosed by 2 membranes, each with a phospholipid bilayer. So it has an outer membrane, an intermembrane, and then an inner membrane, and then an intermembrane space. And again, I just want you to know those key features for now, and we're actually going to see and understand the significance of these various spaces next week when we look at cellular respiration. And then the cristae over here are just the folds of the inner membrane, and then the matrix is the space within the inner membrane. Okay. The cytoskeleton then is basically a network of protein fibers that extend throughout the cell. Primarily, they're there to just maintain the overall structure and support the cell and the activities within the cell. The main types of fibers that make up the cytoskeleton are microtubules, intermediate filaments, and microfilaments. This is an example of a microtubule. Microtubules are straight hollow tubes composed of globular proteins called tubulin. We will see that they play a vital role in cell division later on. I believe that's going to be week 5 or 6. And they help shape the cell as well, and they act as tracks along which organelles with motor proteins move. And this is a fantastic little GIF image that I found that is demonstrating that. So we have here a transport vesicle, for example, here. This is a motor protein known as kinesin, Kinesin, and it is walking along a tubulin track, okay, or a microtubule. So a microtubule is this track here, which has been made up of the protein tubulin. So a bunch of little tubulin balls came together and they generate this track. And this track is what kinesin walks on, and at the same time kinesin carries transport vesicles and other things along this track system. So that's kind of the main feature and what I want you to get out of microtubule. Intermediate filaments are made of various fibrous proteins that supercoil into cables, and their primary role and function is to shape the cell and anchor down some of the organelles. So not all the organelles will move around. Some of them are anchored and will stand still and stay in place within the cytoplasm. Again, this image here just shows you that, again, various fibers of proteins come together. So a single chain of fiber proteins would be a monomer, 2 of those a dimer, 2 pairs of dimers come together to form a tetramer, and then ultimately when the tetramer starts to get knotted and tie up together, it becomes a protofilament, and basically it takes a chain of protofilaments to make up an intermediate filament. Okay, so just understand that basic breakdown. Microfilaments then is another one. You may have heard of some of these microfilaments, such as actin, which act as solid rods composed of globular proteins called actin. So you have G-actin or globular actin, which again are little balls of protein that will come together and form a chain, and this chain is known as actin, which is a type of microfilament. And again, microfilaments form a 3D network just inside the plasma membrane, and they support cell shape, and they are also heavily involved in cell movement. And again, most importantly, actin interacts with a motor protein called myosin, and this interaction between actin and myosin is what causes muscle contraction. So when you're flexing and you're working out in the gym, that's what's happening basically. So here, for example, we have what's called the sarcomere. This is the microfilament structure of your muscle proteins. And what you see here is the thin filament in red, that's actin, and then you have this thick filament here in the middle, which is green. That is your your myosin, your motor protein. And when you trigger muscle contraction, the myosin will walk along the actin track, and it basically will help pull together the muscle. So here's your muscle relaxed up top. Notice these blue bars here, known as the Z-discs, are out wide. And then when you contract, the myosin walks along the actin in one direction on this end, walks along in the other direction, thus pulling together the two opposite ends of the Z-discs closer together. So we have this elongated version where your muscles are relaxed. When activated, myosin walks along the actin track, thus pulling this end of the muscle cell close and this end of the muscle cell close towards the middle. Okay, so actin and myosin. Okay, so that is Lecture 3, Part 2. Study it hard. Obviously, I went into some great detail there about the organelles. Please review it and listen to it over and over again. As always, feel free to contact me for any further questions that you may have. Have a fantastic weekend, everyone. Don't forget your homework assignments as well as your quiz.